Hello friends, in this video we'll learn to solve various types of numerical questions and some logical question on melting temperature of DNA, that is TM value of DNA. Almost in all examinations like CSIR NET, SET, ICAR NET, GET, these questions are asked and many students fail to solve these questions because in these examinations questions are not asked in same pattern. Examiners ask questions in different way and pattern to check the ability of students to think, imagine and answer the question, and this is good also for improving logical imaginative power and also for upgrading the level of education, because we are living in a very fast, intelligent, competitive world. So, thing is that you can solve these questions and qualify these examinations, but for this you should have clear picture of every concept in your mind. So, in this video, I'll tell you various formula, tricks and key points to solve the questions. Please watch my video till the end and don't miss the key points and formula. One more request before starting the question is for the viewers who have not watched my first video on melting temperature of DNA. Please watch that video because basic information is there in that video. If you know those points, then only you can understand these questions well. Link of video is given in description box. So please see that link. So let's have our first question. Question is, what is the melting temperature of a DNA molecule that is 450 base pair long and has 50% GC content? So we'll try to solve it. But before, let's talk about the reactions of students in examinations when they face these questions. Now what happens? One funny thing is that when students face these types of questions, their reactions, their gestures and their postures are very funny. You can see students' reactions and their gestures, posture in the figure which is there on the screen. First picture is reaction of student while reading the question. Second is just after reading the question. Now next reaction is when they read the question and when they try to solve the question. This one is when they try to solve the question. And finally, last reaction, when they quit and feel I can't solve. This is reality, friends. This happens with many students, but don't worry, don't panic and try to solve and understand these questions. So, remember one thing. Whenever you start solving these types of questions, try to understand the question well before solving it. Figure out what is asked, what is given in question, and what's the concept behind, and then try to find a way to solve. If it is numerical question, try to find a formula or a method which suits with the given information. Now, in this question, we'll see what is provided and what you are asked to find out. So, length of DNA is given. It is 450 base pair long DNA fragment. GC content is also given, which is 50%. Means this DNA contains 50% GC, guanine and cytosine. Now you have asked to find out TM value. Now, if length of DNA is given and percent GC content is given and your DNA fragment is longer than 40 nucleotides, use a formula which is there on your screen, that is TM is equal to 64.9 plus 0.41 in bracket percent GC bracket close minus in bracket 500 by length of DNA bracket close. Now, Use this formula only for sequences longer than 14 nucleotides. Remember, this is key point. Use this formula only if the DNA fragment is longer than 14 nucleotides. Now we'll put the values in formula. Now equation will be Tm is equal to 64.9 plus 0.41. Now put percent GC value, that is 50. Put 50 value, bracket close, minus in bracket 500 by 450. Now 450, it is length of DNA. Now first solve the bracket 500 by 450. Now it comes 1.11. So equation will be 64.9 plus 0.41 into in bracket 50 bracket close minus 1.11. Now do multiplication. For this you should know and understand the basic rules to solve equations in mathematics. So we'll do multiplication. 0.41 into 50. Now answer, it comes 20.50. So equation will be Tm is equal to 64.9 plus 20.50 minus 1.11. Now we'll do addition of 64.9 plus 20.50. Now equation answer will be Tm is equal to 85.40 minus 1.11. Now simple equation is there. Now subtract the values. So answer will be Tm is equal to 84.29. It is equal to 84.3. So Tm is equal to 84.3. So your answer is, your Tm value is 84.3 degrees centigrade. So you have solved this question. Now you have Tm value with you, 84.3 degrees centigrade. Now we'll see second question. Now question is, what is percent GC content of DNA molecule that is 800 base pair long and that has Tm 92 degrees centigrade. So remember, this is question asked at the end of my first video. Now we'll solve it. So first see what is given and what to find out. Okay, so length of DNA is 800 base pair. So length of DNA, it is given, it is 800 base pair. Tm is also given, which is 92 degrees centigrade. And you need to know percent GC content. Now in first question you were asked to find out Tm value. Percent GC was given. Now in this question percent GC is not given but Tm is given and you are asked to find out percent GC. So use the same formula as it suits the given information. So Tm is equal to 64.9 plus 0.41 in bracket percent GC, bracket close, minus 500 by length of DNA, bracket close. Now put the values. Equation will be Tm is equal to, again remember this thing, use this formula only for sequences longer than 14 nucleotides. Okay, this is key point, you should remember this one. So put the values and therefore equation will be Tm is equal to 64.9 plus 0.41 in bracket percent GC. You don't know this value, so keep it as it is, bracket close, minus in bracket 500 by 800. 800, it is length of DNA, bracket close. Now use the mathematics rule to solve equation. You should know the rule of mathematics. So we will rearrange equation for solving it. Now take 64.9 on left hand side of equation. So it will be positive in, no, it will be negative in left hand side of equation as it is negative in right hand side. So equation will be Tm minus 64.9 is equal to 0.41 in bracket percent GC, bracket close, minus 500 by 800. So again take this bracket 500 by 800 to left hand side of equation. So it will be positive Tm. So equation will be Tm minus 64.9 plus 500 by 800 is equal to 0.41% GC. Now put the value of Tm, it is 92 degrees centigrade. So 90 Tm 92, put that value, 92 minus 64.9 plus 500 by 800 is equal to 0.41 in bracket percent GC. Okay, you don't know percent GC. So now solve this. Now subtract these two values, 92 minus 64.9. Now it comes 27.1. Now equation will be 27.1 plus 500 by 800 is equal to 0.41% GC. Now solve this bracket, 500 by 800. Now answer will be, it comes 0.625. So equation is 27.1 plus 0.625 is equal to 0.41 in bracket percent GC. Now do addition. Now answer will be, answer of addition will be 27.725. So your equation will be 27.725 is equal to 0.41 in bracket percent GC. Now for knowing percent GC, simple rule is there, divide 27.725 by 0.41. Okay, so percent GC is equal to 67.62. This is your answer. You have solved this question. You use that formula and you find the percent GC. Now let's see question third, which is sort of logical question, theoretical question. So question is, which of the following DNA fragment has the lowest melting point? Okay, now 4 options have been given. You need to choose one out of them. Now think how would you solve this question? No values are given, only you need to choose one option out of those 4 options. For solving this, you need to apply your theoretical knowledge. So let's revise some information related to this question. Now G pairs with C with 3 hydrogen bonds and A pairs with the T with 2 hydrogen bonds. Look at the screen. So as 3 hydrogen bonds are present between G and C and only 2 hydrogen bonds between A and T, so more energy, that is temperature, is needed to break GC bond than AT bond. So GC bond need high temperature, that is Tm will be high than AT bond. So it means the sequence which is having high GC content will have high melting temperature, and sequences having highest AT content among given sequences will have less Tm value. So answer will be D, your option D, because you have asked to find out the sequence which is having lowest Tm value. So if sequence is containing high amount of AT, that sequence it will be having lowest Tm value. So your answer is D. Look at the options, or all other sequences are having high amount of GC. So let's see fourth question. What is the Tm for following sequence? One sequence is given. Sequence is 5' AT GC CG TG CC GC C 3'. Now let's solve this question. First of all, we'll see what is given in question. So look at the question. You will find nothing, neither percent GC content nor something else is given. Look at the sequence. It is short sequence, less than 14 base pair. For these type of short sequences, remember one more formula. One more formula is there to calculate Tm value. Now look at the formula which is there on your screen. Tm is equal to 2 in bracket A plus T in bracket close plus 4 in bracket G plus C bracket close. Okay. Now equation is, now count the number of, now you need to know number of A, T, C and G. So count the number of A, T, G and C in the given equation. So A is 1, T is 2, G is 4 and C is 5. Put those values in the equation. So Tm is equal to 2 in bracket A is 1 plus T is 2 bracket close plus 4, G is 4 and C is 5. 4 plus 5 bracket close. So Tm is equal to 2 in bracket 3. 2 plus 1 is 3 plus 4 in bracket 9. 4 plus 5, 9. So Tm is equal to 6 plus 36. 9 into 4, it is 36. So 6 plus 36. So your answer is 42. Therefore, answer is 42 degrees centigrade. Now you have found out the Tm value, that is 42 degrees centigrade, by using a different formula. Remember that formula. This is important. Okay, so we have solved some questions. So if you like this video, please like and subscribe to my channel. That is